Okay, should we start? We have 22 people in the room. So first, let me first apologize that I have not been able to upload the new lectures. I have recorded one lecture. I will record another one or two today, and I will upload it. I was little distracted with something else. I'm really sorry, but I will do it today itself. That's one thing. So now let us. Can you not? Is it like everybody is having difficulty hearing me, or is it only Akanksha?
Sir, it is fine.
Yeah.
You are
Fine, sir.
Okay. So if you have any question about transcriptional regulation, please ask me. Else, I would like to do something else.
Sir, I was not able to understand the at the end of chipsec analysis after immunoprecipitation, how we will proceed further.
For what purpose?
For such chipsec analysis to know the sequence of
that is not a problem. That is not a problem. Okay, you can see my blank screen, right?
Yes, sir.
Because you asked, so I will tell you. So imagine this is the DNA, okay? For ease, I am only drawing one line, okay? Now imagine that, let us first take the simplest of the cases, that one particular transcription factor is bound in multiple locations, okay? Let us name these locations 1, 2, 3. Okay?
Okay, sir.
Now what are you going to do? You are going to, shear the DNA, and then you will use antibody against the transcription factor and do the immunoprecipitation, right? So your immunoprecipitate has what? Your immunoprecipitate has this little space on the screen is creating a lot of problem. So your immunoprecipitate has what? Has the antibody against transcription factor, and bound DNA, typically approximately 200 nucleotide, right?
Yes, sir.
Now you know how you have created these DNA fragments, right? Like some restriction enzyme or something you have used, right? Or if it is shearing, then it is double like blunt ended, right? And so you know, if you have used a restriction site, you know the staggered end. If you have used shearing, you know it's blunt ended, right?
Yes, sir.
So whatever it is, you don't know their identity, okay? And from each, because you cannot, generally speaking, nowadays people are doing it, and I don't want to comment on that. But generally speaking, you are having few million cells in your when you started your immunoprecipitation, right? So you will have some million times 2 copies of the chromosome. And those many kinds of fragments will be there, each for 1 and 2 and 3 and some 10,000 of those, right? Like assuming that for one transcription factor there are 10,000 binding sites on the chromosome. Now what you will do is you will simply use your adapter, right? And then, you know, like we have learned earlier about NGS, you will have your glass slide where the complementary sequence to the known adapted sequence is already mobilized, and then you put this whole thing in there. And you will create an array, right?
Yes.
And because you know the adapted sequence, then you will be able to sequence it. Then it will be actually like next generation sequencing application.
Okay, so, sir, but it will be the large fragment, exact where the transcription factor is binding.
Yes, yes, that is an absolutely correct understanding. And you know, I am actually in my own lab, I am thinking of taking advantage of that. So let us imagine this. One cell line that I created that unfortunately is no longer working. Otherwise, I would have been on my way. I could have taught you exactly from there. So imagine that this is a small stretch of DNA, say 500 bases. And then you have all sorts of transcription factors bound to it. You know, like if it is really an enhancer, you'll have all sorts of transcription factors bound to it, right?
Yes, sir.
Now, you do not know where the enhancer is, okay? But you have a reason to suspect. If you remember that my the data that I showed you all about BMP targets that are coming up in the first 1.5 hours after addition of ligand, and that had a lot of transcription factors in there. Do you remember that?
Yes, sir.
So imagine that there are 20 transcription factors in there, okay? Imagine that there are 20 transcription factors in there. Now, because they are all coming up simultaneously after BMP treatment, then there is a possibility that some of them cooperate in an enhancer. So what I can technically do, if I have enough money, I'll raise chanda from all of you, then I will do chipsec for all these 20 transcription factors. Okay, now see the beauty. If we take just this one as an example, then sorry, and imagine that I did first a chipsec with this transcription factor. Okay, so as you rightly pointed out, then what will I get? I will get this with this. So the actual footprint and little bit on the side, right?
Yes, sir.
And let us make the side a little longer. Now, imagine that I do the next chipsec with this one. So each one will come down with the actual footprint and little bit on the side, right?
Yes, sir.
Now, first let us imagine in a very simplistic manner that let's say there were only 4 pieces of DNA. Okay, these 4. For each chipsec, you imagine that you got only 1 DNA, 2 DNA, 3 DNA, 4 DNA. So if you have 4 pieces of DNA and they are partially overlapping, you should be able to make a config, right? Do you understand what I'm saying? That you will have this, by sequence alignment, you will be able to do this, you will be able to do this, so you will be able to arrive at a config from this, right?
Yes, sir. Yes, sir.
Now, if I use very powerful bioinformatic methods, and people who are knowledgeable, I am not knowledgeable, then from each of these 20 chipsec, say there are million reads in each. So you will have a matrix of 20 by million reads, right? And if you can do massive scale alignment algorithm, just the way Craig Venter did the human genome sequencing, you should be able to identify contigs that are parts of an enhancer, right? Because
yes,
the transcription factor binding sites will automatically align themselves. Am I being clear?
yes, sir.
So sir, the last part is not very clear.
Okay. So you take, so you understood this part, right? That if I had only 4 pieces of DNA which were partially overlapping, I should be able to align them, right? Like just like you do with what is that thing called?
Short
multiple sequence alignment.
Multiple sequence alignment is a thing. Blast N, you do the same thing, right? And you do it in a multiple for multiple sequences by mega line, right? So now, but this is only for 4 transcription factors, and you had only 4 DNA. That is a hypothetical situation. Now you imagine that you have 20 transcription factors, and imagine that for each transcription factor, the chipsec has 1 million read, right? So you will have a table with 20 columns for transcription factor 1, transcription factor 2, 3, like this, and million rows, right? Now, if you can conduct multiple sequence alignments in all combination, because this is from the same cell type, right? Not exactly the same cell, but 20 independent experiments, same cell type, so same genome and everything. So you should be able to get this kind of contigs automatically forming because of the mega line, right?
Yes.
So that way, the moment you get a config, you know that this config is made. So typically a transcription factor's footprint is around 8 to 15 bases. And the minimum length of chipsec is typically 200. And an enhancer is generally in the range of 50 to 200, right? So if you get multiple, with multiple transcription factors, if the same or similar DNA fragment is coming down, then you know that all those transcription factors are binding to the same stretch, which is your definition of an enhancer.
Okay, sir.
Yes, sir.
That's what I am told. For that I need little money, so आप सबसे थोड़ा ले लूंगा. हाँ? अच्छा. चलो, anything else?
Sir, I have one question from this analysis. Can we, let's say that one transcription factor is coming up with other some activator proteins. So can we identify other activator proteins by this method?
Yes, there is a very good chance that you will be able to. So in the first step of ChIP-seq, you do a mild reversible cross-linking, right?
Yes, sir.
So I do not know about the technological sensitivity, but theoretically, what you will have in your ChIP-seq is that in that piece of DNA, whatever other proteins are bound, like the nucleosome, other transcription factors, other co-activators, they all should ideally remain bound to the same piece of DNA, right?
Yes.
Because of direct or indirect interactions. Now, the question of sensitivity comes that if the amount is large enough to be used for proteomic analysis, then you should be able to identify all the proteins that were part of the immunoprecipitate. But you immunoprecipitated only for your favorite transcription factor, right? So everything else that came down with it, chances are that they came down with it because of specific interaction with either the stretch of the DNA or protein-protein interaction with other with your protein, right? Or combination of both.
Sir, I have another question. So in
Who
the
is this? यार आप लोग थोड़ा video on करो ना. It is very difficult to talk to.
Sir, in the Viva of IIT Kanpur during admission, I was asked that let's take one activator is interacted with another transcription factor, either direct or indirect manner. So then how will I identify whether the interaction is direct or indirect?
Okay. This was fine. So it is relatively simple. If it is a direct interaction, then if you have the two proteins, I'm assuming you know both the proteins. If you take those two pure proteins, incubate them in a tube, then they should form a complex, right? If they don't, but they form a complex when you add the relevant piece of DNA or some other protein factors, then you know that it's an indirect interaction.
Okay, so for direct interaction, we have to add the pure proteins.
Two pure proteins. That is one way of doing it. There is another way, that is you can actually do glutaraldehyde cross-linking and immunoprecipitate one and show that the other is coming down with it. That is no, that will not work. That will not work. That will not work. Then, because it is transcription factor, so it is a little tricky. Otherwise, to show direct interaction, people have often used like is-to hybrid type technique, where you have half of beta gal on one protein and other half on the other protein, and if they interact, you will reconstitute the beta galactosidase activity.
Sir, but that is for protein-protein interaction, no, sir, or like protein DNA.
No, protein interaction. So here the question is also that, right? Transcription factor and an activator.
And an activator, yes.
Yeah, if it is transcription factor and enhancer, then that's a different issue.
So like no, that was protein RNA interaction. No, sorry. I was reading about some methods about protein RNA interaction, sir. So
yeah. There are a whole lot of yeah. There are a whole lot of methods for that.
Yes.
What is it?
Yes, Sandharv?
Am I audible, sir?
Yeah.
Sir, what is the difference between direct and indirect interaction? Sorry, I don't know what that part.
That's fine. Just because you don't know, that's why you are a student. So this will be a direct interaction, while this will be an indirect interaction, right?
There is some kind of linker between the proteins or something.
Yeah, like there is a mediator of the not in the transcription factor sense mediator, but there is a mediator for the interaction.
Okay.
Sir, we can use FRET technology, FRET for the direct interaction of two proteins.
Yes, you can use FRET. Yes, you can use FRET, but any of these methods that will require generation of modified constructs is more time consuming than a straightforward biochemical method.
Yes, sir.
That is the job of a good scientist. For anything and everything, there are multiple methods available. Like, so many times I have corrected you guys that don't propose microarray or RNA-seq for identification of two RNAs. Like, will you not be able to identify? You will be able to identify, but the lab will go bankrupt. So that is the job, that if you have a very thorough understanding of the subject matter and the technology landscape, you should be able to choose which one to use for a particular purpose wisely.
Yes.
Yes, Akanksha?
So I have a question. Chromatin condensation. So from what was written in the book, like chromatin condensation is also a way of transcriptional regulation.
Yes,
so my question.
I will upload 2 lectures today that will cover this part.
Okay, all right.
But you can ask.
Yeah, but so what I wanted to ask is like the pattern of chromatin condensation, is it different in different cell types, and like what governs the same?
Absolutely. Like, many people believe that the differential transcription is a readout of differential chromatin condensation. Okay. Hello?
Yes, sir. So like how the chromatin is wrapped around nucleosomes and all. So what governs that? Are there genes that govern the wrapping and condensation also?
The wrapping seems to be like happening simultaneously with replication, but the getting to higher order of condensation is certainly genetically regulated. Like I will talk about it. There are proteins like the SIR proteins, SIR2, 3, 4, SIR1. These are important, players in chromatin condensation. Similarly, there are proteins like saga complex, etc., which are important for decondensation. But let us not get there today. Let me upload those 2 lectures today, and after that we can pick it up. Okay.
So then we have like another level of gene control, which is the epigenetics. So like acetylation, methylation, that will also govern which genes are active or which genes are not active. So that
Yeah.
also, yeah.
So it is, it is perhaps 2 sides of the same coin, because acetylation, methylation, etc., actually dictate the state of chromatin condensation.
Yes. Yes.
Okay.
Also another question. So, like you had discussed in the slides that because the promoter is like largely invariant, so it must be the enhancer that is governing the rate of transcription and all. So, why in case of bacteria and all, we call certain promoters as strong promoters and certain promoters as weak promoters?
I answered that in the other class. The simple reason is this, that when you see the position weighted matrix data that I showed for the promoter sequence, let me see if I have it. It is just dependent on how much conformity is there. You didn't understand, right?
No, sir.
No problem.
So 30.
Yeah. So you see that there is this 83% probability that there will be a T in the first place, then 91% that there will be an A in the second place, right? So this is statistics, right? As they say that there are 3 degrees of lie, damn lie, and then statistics. Okay. So it does not rule out the possibility that for a particular gene in the promoter, in this location, there is actually a G instead of an A. It is possible, right?
Yes, sir.
The moment that happens, then that promoter sequence is little bit off from the consensus.
Yes, sir.
And because the Pol2 is also invariant, or in reality the sigma factor, or in bacteria, or the TBP in eukaryotes, those are invariant, and those are the ones that are making direct contact with the promoter. So if the promoter sequence is a little bit deviant from the consensus, then it will be a weaker interaction, right?
Yes, sir.
So then that will be called a weaker promoter, right?
Okay, all right.
Yeah.
Sir, also, I didn't quite understand the method of enhancer trap. Like what happens in it?
It is it has been a while that someone has used it again. So imagine that this is everything, huh? Promoter, enhancer, and then this is, say, alkaline phosphatase. Okay, it has, this cassette has everything. Now imagine that this cassette is in the context of, there are some elements that are, I did not have the opportunity to teach you this time because the course is going very slow. Uh, Line elements, etc., which are really remnants of retroviruses that can promote integration in the genome. Okay, or you have some other sequence that can promote random integration in the genome. So now, if you take, like a zygote and inject this, then this construct will be inserted somewhere in the genome, right, randomly. And this way it will be, if you do it in hundreds of cells, and in each mouse this is integrated somewhere, right, this construct. Now if you are looking at early embryo, you can actually do a whole mount development of alkaline phosphatase signal, and say in a particular instance you get the signal in the limb, okay, and nowhere else. So then you know that this construct got integrated close to a sequence which is a limb specific enhancer. In some case, say you got that AP is only expressed in heart, then you know that you are someplace close to an enhancer which is dictating expression in the heart. And then because you know this whole sequence, you can design primers going outward and read it, and you will be able to know the neighboring sequence on either side, right? And then you have a good guess that what can be an enhancer, right?
Okay.
Okay. Okay, चलो अब मेरा बारी है. Now I will go for Umar, for you.
Yes, sir.
Try answering this question. Okay, sorry. Yeah, try answering this question.
So we got a history question. First, light blue color it is exposed to extreme light goes to a state and becomes dark purple and can survive for a long time. This is white straight and the normal. This is straight and the normal circumstances can survive. And enable transcriptions. First. Sir, for the A part, we'll isolate the DNA and add RNA pol 2, purified RNA pol 2. No, okay, just a minute. Just a minute. No, sir, I'm not getting this one.
Anybody?
Yes, sir. Can I answer?
Yes, who is this?
Sir Priyanshu.
Priyanshu, bolo.
So, sir, in the first A part, since the statement is given that RNA polymerase 2 is most sensitive to alpha amanitin treatment.
Yeah.
So if we treat the cells with alpha amanitin, that means RNA polymerase 2 will be not functional or inactivated.
Sure.
So we know that, so in that case, we can then see that if the dormancy is taking place or not and when we expose that organism to 42 degrees Celsius. And if
Absolutely
it is,
correct.
if
Absolutely correct.
yes, for
Yes.
B part, uh, so we will have control in which the normal temperature growth is taking place and at which 42 degrees Celsius growth is taking place.
Yeah.
So we will do microRNA. Since the genome has been sequenced, we can do microRNA
Yeah.
of mRNA, which is like which genes have been overexpressed when the dormancy has occurred.
Yeah.
And
Yeah.
compare it.
Here the only thing is that timing is important. Okay. You should do the microarray within 5-10 minutes of exposing it to 42 degrees centigrade. Otherwise, once it has, because you are interested in genes that are necessary for inducing the dormancy. So if you wait for too long, you may not get the things. Okay, so your answer is correct. Just with addition of time, it will be perfect.
Okay, sir. For question part C, how will we define the part? So we can do RNA in situ hybridization, and we can then check, or
फिर
we
से
can.
पढ़ो, question फिर से पढ़ो.
dormant state. Yes, so like in the dormant state, we can check. I'm not sure, like dormant state, how do we check it? But if the spore has been formed, then we can check if the transcription is occurring in that or not for those genes which we have identified in the part B.
How would you do it?
Sir, we can do an RT-PCR against any housekeeping gene that is known.
So that will not distinguish between transcription that is happening now versus RNA that is remnant. Do you understand what I am saying?
Yes.
Yes.
Sir,
So
we
can't we
can.
use
Sir, we can expose the cell with the radio-labeled nucleotides and for brief time period, and then we will analyze if any radio-labeled mRNA is formed or not
Yes,
by
absolutely.
autorating.
That is the answer. That is the way to do it. Okay.
So adding to Subhajit's answer, like can't we also use antibodies against like the phosphorylated CTD and the unphosphorylated CTD just to like visualize it at the time point, like whether the transcription is going on or not?
Well, yes and no. You are assuming that the CTD phosphorylation status is quickly turned over. That information we don't have in this question, right?
Okay, got it.
But Shubhajit's answer will get you unequivocal answer because if after inducing the heat shock, if any transcription is happening, then it will incorporate radio-labeled nucleotide in the RNA. Right?
Yes.
Last question.
So can I answer?
Yeah.
So for this experiment or for this part, we can do an experiment like we can take a mutant with which does not produce purple color and another mutant that does not induce dormancy. We can cross them with each other, and if we get a wild type phenotype, then I mean we have to do a complementation analysis to get to this answer.
How will you get these mutants?
We will do I mean we have to do random mutagenesis, and we have to design a genetic screen where we have to select for the colonies that do not produce purple color on placing them onto 42 degrees Celsius, and another screen in which we will be selecting for the colonies that do not produce dormant spores. Spore formation is not there on 42 degrees Celsius. Then we will isolate those two colonies and we will cross them with each other. And if we get a wild type phenotype, then this means that they are in two different genes. And if we get a mutant phenotype, that means that they are controlled, both the characteristics are controlled by a single gene.
So, your answer is almost correct. So, if in random mutagenesis screen, you get like either the mutants are deficient in both the functions, that is formation of purple color and dormancy, undergoing dormancy, then you know that the same gene is involved. Like that's a suggestion. But if you get mutants which are sectoring, okay, meaning some mutants can still produce the purple color but cannot undergo dormancy. Some mutants can undergo dormancy but cannot produce purple color. Then this is a very good hint that they are mediated by two different genes, and then a complementation analysis will seal it, right?
Okay, sir.
Okay. So I have a doubt from that C part to determine like whether the transcription is taking place in the dormant state or not. So we are actually isolating the nucleus, right? And then we are performing the assay to see whether any transcript is there, radio label transcript is there or not.
Yeah.
So if it is for a bacterial cell, there will be co-transcription translation.
It is yeast.
No, sir. If in the question, if it
Uh-huh.
was for a bacterial
No,
cell,
no,
then
no. You
no,
see, it is.
no, sir. I know it is. If
Okay,
it was,
okay, okay.
then
If it were a bacterial cell, okay.
yes, sir. Then there will be co-transcription translation.
Uh-huh.
So how do we isolate the, like, there will be no compartmentalization, right? There is no compartmentalization. So how do we proceed in that case to determine?
No, even then, even if there is co-transcriptional translation, even then, if you very mildly open up the cell and add radio-labeled nucleotide, or you simply grow them in presence of radioactive phosphate. Okay, so radioactive phosphate can be taken up by the cell. Radioactive ATP or CTP cannot be taken up by the cell. So if in the end, because for phosphate we have transporters. For ATP, CTP, cell surface does not have transporters. So if in the end you get radio-labeled RNA, that must mean that after the heat shock, RNA has been formed, irrespective of whether protein is also forming or not.
Okay. Sir, one more thing, like for this co-transcription translation, how much stretch of the mRNA needs to be synthesized before it can start translating? Like,
I do not remember exactly, but it is, I think, 20 to 25 nucleotides.
Okay, so like only that much stretch will be detected.
Yeah, only that much. No. It does not have to be detected in there only, right? When I will imagine that when you started the heat shock, a particular mRNA which is 2000 nucleotide long, 500 has already been synthesized when you started the heat shock.
Okay.
If it continues the transcription, then the remaining 1500 in there, you will incorporate some radioactive bases.
Yes.
So it doesn't have to be that part.
Okay.
Fine.
Yes, sir.
So can you please define the time factor that you were talking about in dormant state formation?
Okay, so that you are giving a heat shock and followed by that a dormancy is being established, right? So you are assuming that some transcriptional regulation is in play, right? So
Yes,
now, as we know that transcriptional regulation is always hierarchical, that there will be an immediate response, and then in the first wave, if some transcription factors are induced in the first wave, then those transcription factors can induce transcription of another set of genes, right? And so on. So that way, if you wait for too long, you might end up getting transcripts that are not directly related to the heat shock induction. That are indirectly related. But if you do it within 5-10 minutes, then you will get the immediate respondents.
Okay.
Okay. Let us try this one. New set of people. Ruchira. रुचिरा सो गया क्या?
No, sir, I'm here. I'm reading the question. Uh, Sir, so for the A part of the question, we can expose this W303 alpha strain to this A pheromone, like externally, and in a microarray see which genes are expressed under those conditions, and the one which are normally expressed in a yeast strain, in that particular yeast strain.
Your question is that design a screen to identify the genes responsible for mating response, not the ones to identify the respondents of mating response. Do you understand? There is a difference between upstream and downstream. Here you are talking about upstream. Do you automatically see it, or only because I said it you are realizing it?
I was a bit confused about that actually, because in that it mentioned that it does not respond normally. It does not undergo switch. So I was a bit confused what exactly the A question meant.
See, it is saying that design a screen to identify the genes responsible for mating response in yeast.
Yes, sir.
So without these genes, the mating response cannot happen. And after the mating response happens, another set of genes are transcribed, that is given there, 200 genes transcribed. So what is an easily noticeable outcome of mating response?
So there is mating type switch, or the...
How would you determine mating type has switched? Read it again. Read the second sentence again. Jor se paro.
When exposed to A type pheromone, alpha type S cerevisiae switches on the mating pheromone response. That includes transcriptional upregulation of more than 200 genes and cell cycle arrest. Cell cycle arrest.
So cell cycle error.
Cell cycle
Yes.
arrest should be the answer, I guess.
Yes. So you have to use cell cycle arrest as your assay.
In mutagenesis screen.
Yes. In the mutagenesis screen.
Okay.
So even if you don't know how to assess cell cycle arrest, you say that, we will use a method that is commonly available for determining cell cycle arrest. We'll do this. The rest you know. Okay. In genetic screen, or in, like, if any of you have studied or you will practice protein purification, in both the cases, it is the assay. That's what matters. Rest is routine. Okay, it is your smartness is about choosing the assay, right assay. Okay, like I told you about my favorite assay about how to find the genes responsible for drunkenness. Did I tell you that story?
No, sir.
No, sir.
Oh, this is my favorite screen. So what happens is that if you keep flies in a chamber that is saturated with alcohol, then flies get intoxicated and they fall at the bottom because they can no longer fly, right? Yes.
Yes.
In the bottom, if there is a liquid, then they drown and die. Okay. Now, if you do random mutagenesis and then release the mutant population in the same chamber, then the ones that are mutated for genes responsible for drunkenness, they will not get intoxicated. So they will keep on flying and survive. So the genes, the flies that remained on the top, they are the mutant ones. Okay, so this is how, like, your smartness is about determining the assay. The rest is routine. Okay, chalo. Imagine you have done that. Now go to B.
Sir, one thing here, sir, why there will be a cell cycle arrest?
I do not know why, but it's the natural response.
Okay.
Yeah. Go to
So,
B.
sir, after mutagenesis, will we look for the mutants that will not undergo cell cycle arrest and they will continue
Yes.
dividing?
Yes.
Okay.
B.
Sir, wouldn't the answer again be a microarray? Like, I do not want to say microarray again, but since it's a transcription factor involved, so we can inactivate transcription factor in one case and have it function normally in other, and then see which of the
Question fits a problem. Read it loudly.
imagine then one of the genes you have identified is the transcription factor T. It is also known that many other genes are expressed in S cerevisiae upon exposure to A-type pheromone, including gene G1, G2, G3, and G4. How would you test if this transcription factor is indeed responsible for the expression of genes G1 to G4 upon exposure to A-type pheromone?
Yeah. Is it saying that identify all the genes that are transcribed because of transcription factor T?
No.
Or is it saying just demonstrate that T is responsible for transcription of these 4 genes?
Yes.
Which one?
It is just asking if tree is responsible for the transcription of these 4 genes.
So if these 4 genes are being transcribed or not, how would you determine that?
We can always do RT-PCR for that.
So that's it.
So in one case, we will inactivate the transcription factor and then do RT-PCR, and in another, we'll normally check. Or is that?
You will get 3 out of 10. You will inactivate the transcription factor, but remember that this transcription factor is produced only when the cells are treated with A-type pheromone, right?
Yes.
You do not know for sure that for transcription of genes G1 to G4, T is indeed responsible. There is a possibility that something else called X is responsible, right?
Yes.
And that X is also a slave of treatment with type A pheromone.
Yes.
So you should inactivate T. You will do side-directed, you will do homologous recombination mediated knockout of T in yeast, and then you treat it with A-type pheromone.
Yes.
And in parallel, a normal cell.
Yes.
In the normal cell, if you can detect G1 through G4 by RT-PCR and not in the T knocked out cell, then only you can say that T is responsible.
Yes.
Just knocking out T may not, if you do not get the 4 genes, that may mean that T is responsible. That may also mean that X, which is responsible, is not turned on because you have not treated with the A-type pheromone.
Yes. Okay.
See, ये तो सीधा syllabus है.
I would like to add one more point regarding
Who is this I?
uh, th- this is Nageshwar speaking, sir.
Bolo.
Yes. So to test if this transcription factor T is responsible for expression of genes G1 to G4. So after we inactivate T using after we inactivate T, can we then put another? Can we then design a construct where we induce the expression of T and with the help of an inducible promoter, and then see whether genes G1 to G4 are being expressed when T alone is expressed without any A-type promoter.
Yes, that will be a very good confirmatory experiment. That will be a very good confirmatory experiment. Because in this case, you are bypassing the need of treatment with A-type pheromone because you are putting expression of T under an enhancer which has nothing to do with mating type response.
Yes, yes.
So that would be a very good independent verification.
Okay, sir. Fine.
Now C. मुंह खोले हो तो C का भी आंसर दे दो.
So for this, we can do chipsec as well as EMSA, and we can also do deletion analysis.
Deletion analysis will not be useful. ChIP-seq or EMSA will be confirmatory that it binds within the, like where it binds.
Okay. But
Sir,
why not deletion analysis?
Because deletion analysis, what will be the outcome? What will you assess?
We will assess that after deletion, if what particular construct that we delete, the transcription of G1 to G4 is not observed.
That is question D.
Yes, question D, we can do the deletion.
Yes.
Okay, I did not read question D actually.
बहुत सोच समझ के क्वेश्चन करता हूँ. स्टूडेंट्स को सताने के लिए पूरा पूरा दिन बर्बाद करता हूँ मैं. Okay, so this is how the questions will be. Okay, like integration of all the concepts.
Yes, sir.
Last question, ये वाला try करके चले जाते हैं.
The second question.
Yeah. You know that this is real, right? TBX4, TBX5 example आपको दिया था.
So can I answer?
Yeah.
So for the first part, firstly, we will do a lose it experiment. That is, we will create a knockout mutation of that particular transcription factor that is responsible for four-limb formation. And then we will see if the four-limb formation is not taking place. And then
फिर से बोलो. You will do a knockout.
No, loss of function mutation of that particular transcription factor.
So how would you do loss of function if you don't know the transcription factor?
We do not know. The question says that we know, right?
No.
Okay, it says that there is a single transcription factor. Okay. Okay, we have to identify it.
So can I try the A part?
Yeah.
So we can make a.
Who is this I? अच्छा आप लोग
Sir,
वीडियो क्यों नहीं ऑन करते हो?
Sir, I'm Shweta.
हाँ.
Sir, I'm Shweta. So, sir, we can make a it's as it's saying that there's a transcription factor. So for it, we'll that enhancer, and then we'll take promoter with a reporter, and with the help of this.
So if you do not know the transcription factor, how will you get its promoter, enhancer, all these things?
Okay. Yes, sir. Okay.
लॉजिक को छुट्टी मत देके रखा करो.
So we can do a RNA in situ hybridization.
For
the A part.
For which gene?
For which gene?
What is the question in A part?
How do you identify the transcription factors that are exclusively expressed in the forelimb of a chick embryo? So if we do RNA in situ hybridization, then we can actually but again we should know the antisense sequence of the transcription factor.
No, you should know the sequence of the transcription factor. Then everything else will be fine.
Yes.
So now you realize that you know all the techniques, but you are still deeply confused about which technique to use when.
Yes, sir.
Anybody?
Can we use RNA-seq here? Sir, RNA-seq can be used because there we don't need to know the prior sequence.
RNA-seq, how, what?
Sir, at the like after third day, we can isolate the RNA from the cells and look. But no, but there we have to check it against some control. Sorry.
Yeah.
So you can use like micro array of that third day expression of the third day embryonic development of the chick.
हाँ कैसे करोगे बोलो?
So we can compare the
compare with the database that after getting that which gene is expressed.
Okay. Now read the question again.
Can we do
So we can do a gene expression, differential gene expression analysis between leg bud and the bud which forms the hand.
Wing bud, huh?
and then we can find out that one transcription factor which is overexpressed in forelimb bud but not in leg bud.
Your answer is almost correct. You were just missing one piece of information. Fix the question, Paro.
So again, the time factor is important in this case. We have to do it just at the beginning of the third day maybe.
Yes, unless you mention that time point, answer is incorrect. Okay, so because see, it is given in the question, right? Then also assume that this transcription factor is expressed around the third day of embryonic development. So if you take limb bud around that time point, third day of embryonic development, hind limb bud and forelimb bud, and do microarray analysis with RNA isolated from these two, the ones that are found to be exclusive for forelimb bud, your transcription factor will be within that, right?
Yes.
And then you have to go for a definitive proof. So after you have found out what are the likely ones to be expressed only in the forelimb bud, then you should do RNA in situ hybridization. As I said, that RNA-seq, microarray, these are only suggestions. These are not definitive proofs. Then you do RNA in situ hybridization and see for yourself which one is expressed exactly like TBX4, right? Like only in the forelimb bud, right?
So, but still it would tell us the genes which are expressed is not the transcription factors, right? So how do we identify that?
So when you know the genes, then you can easily do a David or some kind of this online software. Or nowadays, any gene you just do a search on the database, you will find out whether it's a transcription factor or not. That's not a big deal.
Okay.
B.
So for this, if we ectopically express this transcription factor in the leg bud, then if it gives rise to hand in place of, forelimb in place of hind limb.
Yeah, that's what it is. Okay. So actually, if you think about it, these questions are fairly simple. You have been taught everything. It is just that where you are getting the challenge is that you are not used to thinking. You are very good. Like if you did not know my question paper type, then if I asked you a question about write and small essay on how forelimb bud and how hind limb bud is specified in chick, you will be able to reproduce everything that is there on those two slides. And I precisely don't want you to do that. I do not care how much information you can remember. I only care how much information you can process. Okay. Remembering wala undergraduate में खत्म हो गया. Now you have to learn how to think. Okay, चलो चलते हैं.
Yes.
खत्म.
Yes, bye.
चलो, bye. I will upload 4 videos today.
Okay, sir.
Bye.